Just waiting for an okay and we'll get started. Hello, everyone, and welcome to our chat room with Dr. Forget, where he will be answering questions from the audience in regards to his presentation on 3D biofabrication. Thank you so much for your presence here with us today. We're hoping you're enjoying our last day of our Congress. I would also like to give a special welcome to Dr. Forget. It is truly an honor to be here with you today discussing such an interesting topic. It's nice to finally meet you, and I would also like to thank you in the name of our committee for accepting this invitation. It is so nice to have this moment with you. And so before we get started with the questions, is there anything you would like to say?
Yeah, thank you for the committee for inviting me. I hope that everyone enjoyed the talk, and I'll be happy to answer any further questions or clarify any point that anyone from the audience might have, and discuss potential collaboration and so on.
Okay, great, thanks. Okay, so we'll get started with the questions. The first one is from Sibeli. She says, have you conducted tests on mesenchymal cells? Have you also tested other types of cells, such as epithelial cells?
No, we haven't worked with epithelial cells. We have worked mainly with mesenchymal stem cells, so human MSCs, but also endothelial cells as well, but no epithelial cells.
Okay, great. I'm reading. Okay, the next one is from Lucio. He says, hello, thank you for your presentation. I would like to know how you managed to keep the materials you use sterile for cell culture. Good question, Lucio.
Yeah, that's actually, that is a great question. Yes, it's always a challenge. So we have developed different type of techniques to sterilize these hydrogel that are based of seaweed. And so it depends on the material specifically. So the hydrogel, we usually prefer so we can autoclave it, some of them. The one with the peptide, obviously, will have to be processed sterile. So we sterilize them, and then we do all the processing, all the functionalization with peptide after that in sterile environment. But yeah, this is a great challenge every time you work with this type of materials, and especially the seaweeds, hydrocolloid are really can be prone to degradation. But we have developed a couple of other tricks as well to overcome this limitation.
Great. Thank you for your response, Dr. Forget. Contamination is usually a huge problem in our labs as well. But okay, we have another question here from Dr. Luciano. He says, could you share with us an intriguing example of how the mechanical properties of hydrogels impact fate and function in your biofabrication experiments?
Yes, the cell I showed, the endothelial cells, we were able to conduct the experiment in vitro first, and then so we did a screen of different mechanical properties, and these particular mechanical properties that we found out in vitro, when we reproduced this experiment in vivo, we found exactly the same results. So we were really surprised that cells were behaving the same in vitro and in vivo, and we were able to get the same mechanical environment that give us a positive result for this organization of these endothelial cells.
Great. Thank you so much for your response, Dr. Forget. Let me just look at the next question we have. It is also from Dr. Luciano. He says, what is the direct role of carboxylated agarose in modifying the mechanical properties of hydrogels using for vasculogenesis models?
So the particular role is that and what is really important in this example that we show is that when you look at these hydrocolloids, when you want to modify the mechanical property, the only way you can do it is by changing the concentration of the dry mass. So if you change the concentration of the dry mass, then you will change the density of material and so on. And so what we show is that by doing this chemical modification, we can have the same mass of dry materials, the same amount of polymer, but then have different mechanical environments. So that was something that was really important for us, because then when we have the same amount of polymer, then the viscosity do not change, and that's really important for the bioprinting process, because before, when we do the extrusion, the viscosity of these materials are the same, but then the resulting hydrogel have different mechanical properties. And so depending on the amount of carboxylation on this polysaccharide, we can, with the same viscosity, same amount of material, generate hydrogel with different mechanical properties. And that's really a key features of this material, is this that we have the same amount of polymer, same viscosity, and so for processing is really a key. And also we do think that this has also some biological impact, that where we have just one parameter that is changing is the mechanical properties, and not for instance, in alginate, that would be the amount of calcium or the amount of polymer. So here we just have the same amount of polymer, and only the amount of carboxylic acid is changing.
Great, thank you for your response, Dr. Forget. I just wanted to say that we have a full chat room, and everyone is complimenting your work and your presentation. Ulysses says, Buenas tardes, excellent lecture. Gabriel says, hello, thanks for the lecture. It was very good. I agree with both of them. Thank you so much. We still have a few minutes left in our chat room, so I guess we can just wait a little bit for any further questions.
I have more comments as well on that. So, so we are really, I was really keen of participating in this conference, because we work with these seaweed extracted materials, and we are really keen on exploring the use of other seaweed extracted materials. And I know that in Brazil you also, and especially maybe in Embrapa, you might have some connection that could help, be helpful for us to connect us with scientists that are working with seaweed, and that where we could get samples, try to extract new materials, try to see what we can do with this seaweed for biomedical application, of course. And so we're really keen. I know you have also some sargassum issue as well in Brazil, and we would be also interested to work with people working on this topic. So I don't know if anyone has connection in Embrapa or is working in this direction. I will be really keen on being connected with these people.
Great. Thank you so much for wanting to work with us. I'm sure Dr. Luciano knows many people who will be glad to help. I'm working on vegetable extract on my master's now, and he has many connections, so I'm sure he will be in contact with you to make this happen. We have another question for you from Dr. Luciano. He says, we are curious about your card game. Yes, I saw that card game as well.
Oh.
It was so nice. I really liked it. I was like, what a nice way to learn chemistry. It was nice.
Yes,
yeah, go on.
That's a fun game. That's a fun game that's developed during the first lockdown in COVID, and now I've been able to commercialize it. I'm not sure I'm able to ship to Brazil, but I have to have a look at that, how we can send some
Oh,
to you guys. And but yeah, it's a really fun way to teach chemistry, and it's been used as well in high school for the last year of high school. Because instead of going down into the detail of chemistry, which can be quite difficult with all the math and so on, then you can just play a game and have the first concept already locked down before going to university.
of course. Learning the fun way is the best way. Let me just continue
Yeah.
with let me continue with his comment. He says, "We are curious about the card game on your website that teaches free radical polymerization. What motivated you to develop such an innovative and educational tool? And what do you hope people will take away from playing it? Have you ever considered creating a similar educational tool specifically for the field of biofabrication?"
For biofabrication, I don't have anything in mind right now on what could be the best game. But the free polymerization card game is that I found out that a lot of students have difficulty of understanding this high molecular weight polymer and how it is working and the role of radicals. And so the game basically it's to keep the radical alive, and if you keep the radical alive, then you can still polymerize your chain. And so the game is kind of protect the goal of the game is to protecting this radical, and then to play with your opponent and see how he can move strategically to protect his radical without you interfering with the growth of his chains. And so it's really for the student to understand this concept of radical and what is important, why it is important to control this radical, and how you can, and what are the tools that we have as chemists to protect this and grow the ultimate polymer with the highest molecular weight possible.
Right. Thank you. Congratulations on that. Really. We have another question from Henry. He says, "Good afternoon. First, thank you for the lecture. It was incredible. My question is, could we use these hydrogels to study the application of medications on tumor cells? Maybe print the cells next to a tumor a conformation and study the medication application for that?"
For the endothelial cells,
Yes. Yes.
yeah, so that's so we haven't been there yet because it's always complicated to move to a two type of cell system, which is way more complicated than just a single cell system. Of course, something that was in our mind, but again, the goal is to have more people, and sometimes that's might be challenges to do to realize all the ideas we have. But definitely the idea of this hydrogel is also to become an alternative to matrigel, because all of these tests are usually done on matrigel or collagen. And so actually in the paper that we published a couple of years ago, we compared the result of these endothelial cells in vitro with matrigel and collagen and with our material to show that we have really a different type of organization. So in our hydrogel, we saw that we have polarization of the cells, whereas in the other hydrogel, collagen and so on, we didn't have that, and also in matrigel as well. And clearly matrigel, you have a different type of organization of the cells. And of course, in matrigel, you also have the issue of the reproducibility sometimes from the different batch that you might get.
Right. Thank you so much for your response once again, Dr. Forget. I believe we don't have any more time for any other questions, but it was very great. It was very enriching having this talk with you. Before we finish, is there any last words? I would also like to thank you again for being here with us today.
Yeah, thank you for all the great question as well. It was really nice to interact with the audience. And yeah, I'm looking forward to see if any connection with the seaweed people might be coming up next. Thank
We
you.
will keep in touch, Dr. Forget. We will not forget that. Okay. Okay, thank you so much, Dr. Forget. See you next time.
Bye-bye.
Bye. Um, thank you so much, everyone, for your patience and time here with us. Up next, we'll have the presentation of Dr. Balak. So I will see you guys at 3 o'clock. Muito obrigada pela presença de todos vocês. Espero vocês na próxima palestra. Tchau.